Good afternoon, everybody. So I'm Mustafa Mir, and as Gabby mentioned, I'm a PhD student in the QLI lab. And the goal of my talk here is basically to give you guys a feeling for how you go from, how do you leverage the SLIM instrument and all the surrounding technologies that are available to us to actually perform a study and get a biological answer to a real biological question. And so before we get started, I wanted to put in context of other imaging modalities where SLIM lies in terms of temporal and spatial scales. So this is a sort of famous picture by, that was in a review article by Roger Sheehan, the Nobel laureate who won the Nobel Prize for GFP. And basically the way you read this graph is that on the vertical axis over here, the largest is the duration you can image, the longest experiment you can do, and the smallest is the smallest time between two events that you can measure. So SLIM we can measure from a millisecond up to several weeks, as Gabby already mentioned, and our lateral dimension is of course limited by, on the short end by diffraction limited resolution, and on the long end by, as we showed, by stitching together several fields of view. And on this graph, it's the thickest sample you can measure reliably and the thinnest sample you can measure reliably. So here we're from 1 nanometer up to 1 millimeter for SLIM. So this gives you an idea in context of other modalities such as AFM, MRI, OCT, etc. So the point of showing the slide is that really we have a huge dynamic range in terms of our temporal and spatial ranges. And what that really enables us to do is to image a system. And by what I mean by a system here is a biological system. So to truly gain an understanding of how a biological system is working, you need to be able to look at how it interacts or how it evolves on the population level, how single cells are behaving dynamically, how the subcellular structure is changing in 3 dimensions, and of course you need specificity and you need dynamics. So this slide basically shows that in SLIM we can kind of look at all these different temporal and spatial scales to really try and understand what's going on in a biological system. And of course, to image such biological systems, we need to leverage something that's very important in our lab is our interdisciplinary collaborations. And the results I'm going to show you in the talk today are mainly in collaborations with people in the cell and developmental biology for the cell growth, in physics for the E. coli, and we have a collaboration with Professor Stice at UGA for stem cells, and Dr. Tangela at Provena for the blood, and in our own department of ECE for image processing, we have Professor Mindo and Dr. Babajan, and of course our funding agency. So this is really, all of the studies here wouldn't be possible without 2 things. One, we leverage the existing technologies that we have. So we have the SLIM instrument, the Zeiss microscope, which has the nice incubation and all that. And then we leverage the expertise of other people on campus to actually be able to perform these studies. So the first thing I want to talk to you about is blood screening, which was my first project in the lab as well. And as Gabby already mentioned, blood cells are an ideal test bed for many imaging modalities for the simple reason that they're basically bags of hemoglobin. So from the perspective of a quantitative phase imaging microscope, all we have is a constant refractive index and some thickness that's changing. And so it's a very good system to test out image analysis ideas and to test out your system. So Gabby talked about red blood cell dynamics. And he said that the flexibility is very important, how their mechanical properties are very important in their function to deliver oxygen. And the other take on this is that also the shape is extremely important because it's optimized already for squeezing through really small capillaries to deliver oxygen. And it also provides a lot of diagnostic information. So for example, these teardrop-shaped cells called darocytes are associated with thalassemia. These bumpy cells called echinocytes are associated with liver disease, etc. So you get the idea. And the current way that this is performed in the clinic is that, first of all, when you walk in, you get a blood test. The doctor orders a complete blood count test. And what that means is they take a milliliter of blood and they feed it through a Beckman Coulter counter. And that basically tells you statistics on the volume distribution and cell counting, etc. This technology is over 50 years old. It doesn't give you anything at the morphological, at the single cell morphology level. It's expensive in cost and also costly in terms of the human resources and expertise that are required to operate this machine. And what happens is in the distribution you'll get some outliers, and if the distribution width is too wide or is not normal, what that does is it raises flags, and then a technician or the pathologist sits down and scans through the slide and looking for weird cells like this, and it's a manual cell count. So our idea is that we have a quantitative phys image which we can take these quickly, and we can get high throughput by scanning and stitching a large number of these images. And in each image, in each of these large images, we can actually still maintain the high resolution. So now we have quantitative information on single cells, and we have the shape information. And since our signal to noise is really high, we can do image segmentation very easily. So the idea with image segmentation is, if your signal is, if you do a histogram and your background lies over here and your signal lies over here, you can just take the signal and you know where the cells are versus the background. So we can identify all of these cells, thousands of cells in a large blood smear very easily. And now, so the question is, what can we do with this information? So we were talking about the shape. And as we mentioned, in the blood cells, it's very easy to get the height if you know something about the hemoglobin concentration. So this, the definition of the refractive index in a living cell was, it can be approximated by this equation, basically, this relationship which was first realized in the 1950s when phase contrast was first invented. And you can basically approximate the refractive index inside the cell as this refractive increment constant called beta, which tells you how the refractive index changes given the concentration of a protein. The concentration of the protein, which is in this case hemoglobin, plus the refractive index of the cytoplasm. And we know the refractive index of the outside solution, whether we make it ourselves or it's the plasma or whatever your buffer solution for the red blood cells is. So now if we know this refractive index, which isn't trivial by the way, and we know the phase, we can calculate the topography of the cells. We get a height map. So for each cell, we know the topological information. And from that, we can do some simple geometry, get the volume, get the surface area, and then we can begin to think of parameters that are actually relevant to the pathologist. So like we mentioned, like I mentioned in the beginning, we have all these weird shapes. So one thing that can give information is the sphericity, which looks at basically the ratio of the volume and surface area, and on a scale from 0 to 1 tells you how spherical the cell is. Then there's this minimum cylindrical diameter, which predicts how small of a capillary that a given red blood cell can squeeze through given its surface area and its volume. And so, and on top of these, I think we have 19 parameters. So we have a huge, very large parameter space for each cell. So we can think about maybe separating normal cells from disease cells based on all this large number of parameters. And to begin to test this idea, whether this was actually useful in the clinic and whether our volume measurements actually tied up with what's measured in the clinic, we did a small trial with 32 patients in Purina Hospital across the street. And the basic idea was that we would get the volume measurements from the Colder counter and compare them with the volume measurements that we have from our quantitative phase imaging, and see if there's an agreement, and then explore the pathways. So from the Colder counter, all you get that we can measure is the volume and the distribution width of the volume. And we want to make sure those tie up, and then we want to look at all the other parameters that we generate, like the sphericity, the minimum cylindrical diameter, surface area. And really the question is that whether we can use all these extra parameters to avoid intervention from the pathologist. So right now, when the Colder counter detects an abnormality, the pathologist goes in and looks at it. Whereas if we can provide quantitative information on all these abnormalities, then maybe we don't need intervention at this stage from an expert. So here is the result from this study. So this is an example of a sphericity distribution. from a normal patient. So you can see that even in a normal patient, you have cells that go from very flat to the normal discocyte shape to very spherical. And this is an example of a data set that wasn't possible before without this technology, because basically this information was provided only qualitatively. So you would get a volume distribution. If they noticed that the volume distribution was abnormal, the pathologist would go and count the cells in a grid and go like, oh, it looks like there's a higher number of spherical cells. But this quantitative information on the distribution of sphericities is a very unique information that we can provide. And it may actually tie into, looking at early diagnostics for disease, monitoring effects of drugs, etc. And here's another example. This is just looking at the volume distribution, and we show that it ties up very nicely to what we measure with the Coulter counter. And this is a comparison of the volume measured with our technique, which we call diffraction phase cytometry here, and the CBC, which is a complete blood count given from the hospital. And what you'll notice over here, so there's two data sets plotted here. One that says raw, which shows the poor correlation, and one that says corrective, which shows a high correlation. And what we did over here was basically, in the raw, what's showing is that I assumed that there was a constant, that every patient had the same hemoglobin concentration, which is obviously not true, which we realized very quickly when we plotted this. And it was taking into account the hemoglobin concentration measured per patient by the complete blood count analysis. And once we took that into account, we had very good correlation with the clinical results. And what basically that taught us was that we have to be very careful about this, and we have to measure the hemoglobin concentration ourselves, and if possible at the single cell level. And so there has been a lot of effort in that, and I won't get into too much detail, but there are several approaches, and the main idea is to decouple. Okay, so when we measure the phase, what is the phase? The phase is the product of the refractive index and thickness, right? And we need to measure we need to have enough measurements to basically decouple those two unknowns. So one way is to measure the phase at several wavelengths, and then the other one, which I worked on last year, was to measure an absorption map and a phase map. And basically in the absorption map, you just follow Beer's law, and you have some attenuation given the concentration. Here you have a change in optical path length given the concentration and the thickness in the equation that I showed you earlier. So now you have two equations, two unknowns, and you can decouple the hemoglobin and the thickness information that way. So this is just a small example of like bringing in a different measurement to complement your phase information. So the idea of showing you this blood testing, like you can think of it as a case study, was that this is a very kind of illustration of how you can go, when you have quantitative information, you can do simple analysis and go a long ways. Like all we had was, we have a blood cell, which is a simple model. Now we have quantitative phase information, and all I'm doing is looking at simple geometrical parameters. But since the information is so new and so novel, it can teach us a lot more than just looking at a blood smear. So now I'm going to get into something which I think is far more interesting, and that's looking at mammalian cells, actual mammalian cells, not red blood cells, which have no nucleus, and they don't grow and divide. They're made, and then they go for 90 days, and then they get degraded.
They're very nice.
They are very nice, but so when you think about, let's think about developmental biology, and what I'm showing here is a picture of an embryo that's going from 13 days, 13 weeks to 23 weeks, I don't know, but it shows, not weeks, but it shows what goes on in the development of an embryo. And there's three main processes that happen in developmental biology: growth, differentiation, and morphogenesis. And by growth, I mean that the cells are growing in size and they're dividing, so they're growing in population and in size. Differentiation, that you're going from these embryonic stem cells to a huge variety of different cell types. And by morphogenesis, what I really mean is motility, that you have these different types of cells, and now they have to arrange themselves spatially to actually form organs and to accomplish their tasks. And what I want to talk to you now is that we can use SLIM to measure all these three different processes. So remember in the beginning of the talk, I spoke about the idea of imaging a system. And what I want to show to you is now that if you leverage all this existing technology, that it really is possible to look at these three, like basically cornerstones of developmental biology. So first, let's talk about cell growth. And this is a nice graphic which shows these human osteosarcoma cells, U2S cell line, that goes, that you can see these two cells that are sitting side by side that are growing. And the thickness or the color here is basically corresponding to the density at each pixel. So cell growth, or measuring cell growth, has been defined as one of the last big unsolved problems in cell biology. And the reason why this really is the case, and the reason why this is difficult, is simply because cells are really small. We were talking about on the order of picograms. And on top of that, there's not much change to work with. They only, typically cells double their mass and then they divide. So you go from 1 picogram to 2 picograms back to 1 picogram. So you have to really, to actually even answer simple questions such as whether this is growing exponentially or linearly, you have to measure with a femtogram level accuracy. And on top of this challenge that they're really small is that cells come in all sorts of shapes and they like to move around. So it's a big, huge engineering challenge. And there have been several attempts to kind of address this. And the earliest one is, again, the Beckman-Coulter counter, which has been used for the last 30 years to answer this question. But here you're basically just measuring population level statistics. And you look at the volumes of a population of cells, and then you look at, wait 5 days, and you measure the volume again, or you add some drug and you measure the volume, but only population level statistics. Second is if you assume, if you have a simple cell type like a yeast or E. coli, where the geometry is constant, you can just go from phase contrast of fluorescence and assume that the size is proportional to the mass. That means the density is constant to the cell's life cycle, and get growth information that way. We know that this isn't that reliable. The second class of methods basically is using MEMS technology. And there has been work at MIT in which they basically have this cantilever system, and as it has some resonant frequency, and it's in a microfluidic channel, and as you flow cells through this, it changes the properties of this resonator, so the frequency shifts, and you can get the mass from the shift in the frequency. And the second is this kind of, this one is limited because the cells are actually flowing through the system, so you can't follow one cell over time, but it's pretty sensitive. The other one is to actually be able to follow a single cell over time, is this pedestal-based system that was developed in Professor Bashir's lab across the quad. And in this one, they can measure a single cell over time, but now they can look at, they can differentiate it between two cells that are growing on a pedestal. They can look at, and the sensitivity is actually decreased as compared to this method. So even in these papers, if you read through them, in the introduction, they usually mention that the ideal method would be an optical method, in which we're minimally invasive, and we can follow cells and their progeny over several generations, and actually be able to look at different cell types and not mess with their microenvironment too much. So as we mentioned, SLIM, if you translate those nanometer path length sensitivities to dry mass, we have a 1.5 femtogram per micron square sensitivity to dry mass, basically. So this is sufficient to measure cell growth. And of course, to actually do these experiments, we need to leverage the environmental control technologies that have been developed by Zeiss, so we can maintain the temperature, the CO2, and the humidity for up to a week, or actually we've done it for 2 weeks now, and keep the cells alive and happy and image them continuously. So this is the movie that you saw earlier of the cells growing over a week, and I'm just showing a snapshot of 4 hours and 45 hours. And this is at the highest level, at the population level. And what you're this was a synchronized cell culture, so all the cells are supposed to be in the same cell cycle phase. And what and the red graph over here is the mean cell dry mass, so the total mass divided by the number of cells, basically. And the black line is the total dry mass of the whole field of view. And what you see is a deviation between these two curves at 24 hours, which kind of is meant to illustrate the problems with trying to study cell cycle behavior in a synchronized population, is that over time they fall out of synchronicity. So you're not really getting a pure cell culture that's in G1 phase or S phase or G2 phase of the cell cycle. So it's important to be able to look at each single cell and say what phase of the cell cycle it is in. And of course, then you don't have to chemically or mechanically control which part of the cell cycle the cells are in. So this is an example that is meant to show that we can track single cells. And the idea here, we went to E. coli cells first because there is a nice simple model organism and it's well studied. And so this is the dry mass density map, or the SLIM image. And the black line corresponds to this cell. The red and green lines correspond to its two daughter cells. So it's meant to show that we can follow a cell, and as it divides, we can follow its daughters. And in this system, we had a sensitivity of 1.9 femtograms. And we can measure several families like this, and then look at the overall behavior of those cultures. And we found that the growth rate versus mass is linear, meaning that we have exponential growth. So this, might not be exciting to you, but this was something that was impossible to do at the single cell level until using something like a microscope, until SLIM was invented because of its spatial sensitivity. And this agreed very well with the measurements that were done with the microresonators. So now we wanted to do something a little bit different. We wanted to look at something that microresonators cannot do. And that is to actually look at the growth in each individual phase of the cell cycle. And so to accomplish this, we leverage the multimodal capabilities of the system. So because we have fluorescence, the fluorescence channel available to us, along with the SLIM, we use this PCNA marker, and which was being transfected the cells with this. So the PCNA was tagged with YFP. And now in the S phase, I don't know how familiar you guys are with biology, but mammalian cells usually have a G1 phase, S phase, G2 phase, and then mitosis. So they're growing in the G1 phase and the G2 phase, and in the S phase they're supposed to be replicating or synthesizing the DNA. At least that was the classical view. And so our idea was, how does the growth actually change in these at the single cell level? So as you can see, the PCNA, the spatial organization of the PCNA is very distinct in the S phase. So if you have this information, we can save when the cells in G1 versus S versus G2, and then we go ahead and measure our mass along with this fluorescence, and now we can split up the curve according to G1, S, and G2. So again, this is not only this is an example of how you can leverage existing knowledge to complement your quantitative phase imaging. So we have this great quantitative phase imaging instrument, but would we have been able to do the study without actually having, leveraging fluorescence? No. So it's very important to, consider other technologies when you're designing your experiment and when you're trying to answer a real biological question. And because of this, we were, for the first time, able to actually measure single cell growth according to the cell cycle phase. And we found in this that basically, contrary to the current knowledge that there's constant exponential growth, we found that there's different types of growth in ech phase, and we found the highest growth rate, exponential growth rate, was in G2. So now this was a nice basic science study where we wanted to look at how the growth rate is changing in different parts of the cell cycle. Now to move forward, we want to actually measure something that's also clinically relevant, and to actually modulate the growth rate and see what we can, with the effects of different drugs or the microenvironment. So right now we're working on a breast cancer model using MCF7 breast cancer cell line and looking at the effects of estrogen versus non-treated. So what estrogen is supposed to do in breast cancer is really increase the proliferation rate, so they grow a lot faster. But as this has been known for like 40 years, 50 years now, but nobody has actually looked at it at the single cell level. Nobody actually knows what happens at the single cell level when you administer estrogen, and even the temporal resolution is limited to days, and that's because they typically use these MTT assays that looks at the metabolism of a whole well rather than looking at the single cell. So now using SLIM, we can actually go in and look at single cells and see how the response to estrogen is. And our hope is that this will teach us a lot about the actual basic level growth behavior of breast cancer cells. So this was just a sneak preview. And now we'll move on to the next topic, which is cellular motility. So as I mentioned, the three cornerstones of developmental biology. So this is related to morphogenesis, or actually after the cells grow and they differentiate, how they organize themselves is by being mobile. And so here I'll show you examples from two quick studies. So as you know, in our images, in these images, we not only have the mass, we don't have the density map, we actually have the whole image, and we can track the cell over time in that image, right? So in combination with the growth and the mass and the structure, we can actually measure its motility. So this is showing the X position and the Y position of two different cells. And in this first study that I'm showing you, what we did was that we had Drosophila S2 cells, and we attached them to a glass cover slip that was coated with polylysine. And we found that the attached cells basically don't grow as much as the motile cells. So these cells really don't like being attached using polyglycine to the substrate. And we found a very nice trend in going from the first generation to the third generation in terms of the mean square displacement and the growth rate. So basically, the more that the cells were able to move, the faster they were able to grow, which was basically, it was kind of new because people use polylysine all the time to culture their cells. And if this is the case, that the growth is inhibited so much, that's something you shouldn't really do. Now something a little bit more exciting in terms of the motility is that this is, you're looking at a movie of clusters of neurons that were frozen, and then we put them on a plate. And then we image them for 24 hours. And we watch how they're forming the connections, basically. So when this movie loops around, what you'll notice is that originally there are just simple clusters that aren't connected. And as time goes on, the clusters send out extensions towards other clusters. And by the end of the movie, almost all the major centers are connected with these dense networks of neurites. And now this is obviously not at random, because you can see that from the beginning of the movie, you can see that the growth is happening in the direction of other clusters. So the cells are actually following some sort of chemical gradient in how they're sending out these neurites and how they're actually moving around. And by having the capability to track them and measure their mass dynamics at the same time, we can say something about how neurons like to spatially organize. So again, this is another teaser. This is a work in progress, so I'm not going to tell you anything about how cells are actually organized. And now the last cornerstone is differentiation. And so in this movie, what you're seeing is human embryonic stem cells that are going from the neural progenitor stage to fully differentiated neurons. And you'll see at each day they become more and more neuronal. Sir?
Did you get something to the cells in order for them to differentiate?
Well, you actually remove something to make them differentiate, but that's it. So it's just a different growth medium. So there's a certain growth medium in which they don't differentiate, and then you remove a chemical and they tend to differentiate then to neurons. But so let's watch this one more time. So you'll see that originally they have kind of these wide extensions, lamellipodia, and you see a lot of dynamics in the nucleus. And slowly these things get narrower and narrower, and you see more interconnections between the cells. And now you begin to see transport down these long neurites. You can actually see stuff moving in between the cells now. And this is beginning to look like a neural network. Now we have a neural network. So this is one culture that was sitting on the scope for 2 weeks, and we kept it alive and imaged it every day. So we wouldn't have been able to do that without the environmental control and the stability of the stem system. So but now how do we understand these changes? And the idea is that we want to basically see what is the difference in the dynamic and mass signatures of these cells as they're differentiating. And so as Gabby talked about earlier, when we measure the fluctuating phase signal, there's two things that can cause the fluctuations: variations in the refractive index and variations in the height. And if you measure at really short temporal scales, if you look at things that are moving really quickly, what we're mostly measuring is these fluctuations of the membrane. Now, if you measure on the order of like minutes, or if you're measuring like at a rate of half a hertz, like those previous movies, now these fast membrane fluctuations, they'll average out because they're moving really quickly. And now what you're measuring is these slow kind of dynamics of mass being transported in the cell. And so Gabby talked briefly about the dispersion DPS method that we have. And basically this is the graph to note that in a living cell, this is basically kind of what we expect. That at short spatial scales, we expect the behavior to be very diffusive, because cells don't want to consume energy when they can just rely on diffusion to transport things at short spatial scales. Now, if you're talking about longer spatial scales, you can't rely on diffusion because it'll take forever to transport something. So now you have active directed transport, which is represented by this linear dispersion relationship. And as examples, these are 3 snapshots of different regions in the cell, basically. So this is a microglia in a glial culture. And if you look at the cell membrane, not in any particular direction, just in the membrane, you'll see that it's almost purely diffusive, like if you're not looking radially outwards. And that's because in that area, everything is moving around diffusively. Now, if you begin to look radially outward from the nucleus, you'll have both a directed transport component and a diffusive component. And if you look at actual neural process in which it's very one-dimensional and the neurites are actually communicating with each other, now you see that it's very directed. You have almost a purely linear relationship. So you can have these different regimes in the cell. And the reason I'm showing this to you is that if you remember in the Differentiation movie, in the beginning we have these kind of blobby looking cells with these wide lamellipodia, and they're going into sort of a network that's defined by almost one-dimensional processes that are connecting each other. So if you're trying to say something about a stage of differentiation, maybe this is a good way to go about it. So as it becomes more and more directed in certain directions, then we can maybe say that the cell is 13 days differentiated, or maybe it's 6 days differentiated based on the signatures. So that's the idea, that we can go to each day of the differentiation and look at this dispersion relationship, and maybe we can develop signatures to say how differentiated a certain cell is. So that's one of the goals with the study, and still a work in progress. Okay, so now I'm going to change gears a little bit again and talk about one more application, which is subcellular structure. So we can look at these dynamics and we can look at growth over days. We can look at short dynamics, is changes in the mass over minutes. But we know that the function in biology is intrinsically linked to structure. That's how proteins function. They're very dependent on the structure. That's how the brain works. It's all dependent on structure. So it's very important to look at structure in biological objects, not only in 2D, but also in 3D. But often the problem is that these structures of interest are below our diffraction limited resolution. And so we have all sorts of techniques that claim to kind of go around the Abbe's, or they claim to beat the Abbe's diffraction limit, but actually they go around it by localizing fluorophores. So the main idea in all these techniques, STORM, STED, is basically to turn fluorophores on and off. And if you, either in a defined way, like in STED, or in stochastic way, like in STORM, and if you have signal from only one location, you can say very accurately where that is, and then you can reconstruct a high-resolution image. And you have other techniques that are like structure illumination, and 4-pi, and deconvolution. Well, I'm going to run out of battery, I guess, so let's see. No power?
How many minutes do you have left?
I can finish pretty quickly, I think. It'll be okay. So what I was interested in looking at over here is E. coli subcellular structure, which has previously only been studied using these fluorescent super resolution techniques. And to actually study these structures using fluorescent super resolution, you need to come up with either specialized strains, or you need to come up with okay.
2 more hours.
So to actually do this kind of study in E. coli, they had to come up with different strains. They had to come up with use very complicated methods. So it's not really something you can just do in your lab. You get a wild type E. coli strain, and you want to look at how its subcellular structure is changing. You can't just do that. So we wanted to show that it's possible to do this type of study in a label-free manner. And what we exploited was that SLIM has very good signal to noise. And because of that, you can employ very robust deconvolution algorithms. And I'm not going to get into the details of the deconvolution that we used over here, but basically we were able to get a 2.5 times resolution increase in the lateral direction and 3.4 times resolution increase in the z direction. And you can see basically the change in the point spread function becomes a lot smaller. And using this deconvolution on E. coli, now we can begin to resolve these subcellular structures. And as you can see in these old fluorescence data, that these structures follow these kind of helical patterns. They're all coiled at different periods. And this is very interesting on its own. Like these proteins, these MEN proteins, what they do is they figure out, they tell the cell where to divide. So the cell has to divide right in the middle of the cylinder. And the way they accomplish this is that they shoot back and forth from end to end of the bacteria, and where the concentration of these proteins is lowest, that's where the cell is going to divide. So it's like a really cool evolutionary solution to like very accurately split the cell in half by shooting something back and forth. And it turns out these kind of travel on a helix. Then we have these MREB proteins that sort of function as structural support. These are like can be considered a cytoskeleton type thing for the E. coli, which is not something that's really was expected, but it exists. And we even have helical deposition of lipopolysaccharides and other proteins on the membrane. So in this cylinder, a lot of things are lying on this kind of coiled in this coiled pattern. And in our label-free study, we were able to visualize several of these coils in different cells, and you can very clearly see that this is some sort of helical pattern. And when we looked at several different cells, we found that there's two dominant structures, one which period remains constant as the length of the cell is increasing, and one which is increasing with the length of the cell. And so this was not really possible without employing all those complicated super resolution techniques or creating specialized strains for looking at the structures before. But of course, our shortfall here is that we don't have specificity. So we can tell you that we have these two structures here, but we can't tell you what they are unless we're able to tag different proteins, and either by a process of elimination or by actually getting it right on the first time, tell you what the structure is. So it's again over here, it's important to, at least for one time, to use the specificity provided by fluorescence to tell us what we're looking at, and then we can go and look at it in a live culture without worrying about it. So that's the thing. We need to do one control study with the fluorescence to figure out what the signature is in our phase image, and then we can do it without having to worry about without worrying about the fluorescence. So this is the conclusion in terms of live cell imaging using SLIM, that we have label-free quantitative data, femtogram sensitivity to dry mass. We can look at structures, subcellular structures to entire dishes, And we can look from these really fast short-scale dynamics to actually watching the cell grow over days. And we have this dispersion technique to quantify dynamics without actually having to go in and track cells. And multimodal, fully biocompatible, and as I've shown you, we've looked at E. coli, we've looked at cancer cells, we looked at neurons, so we can look at all different types of cell types. And to kind of set you up for what at least I expect to happen with my own research in the next few years is to actually, first of all, leverage fluorescence technology to gain specificity for both, for actually looking at the synapse formation like Gabby mentioned in neurons, and to look at, get specificity for the small dynamics that we're seeing in the cells. So if you can actually say this is the mitochondria or this is that, we can use that to actually add more biological conclusions to our analysis. And in terms of the signatures, I think there's a lot that we're going to see coming out of this in the next few years, is that each of these cells have their own unique signatures in terms of those dispersion relationships, in terms of morphology. So it's a huge parameter space. And if you can quantify as much as possible about the cells, we should be able to separate two populations even if they're very close to each other. So if you have these cells that look almost the same, but the dynamics are a little bit different, we can tell them apart, say like a cell that's differentiated 5 days versus a cell that's differentiated 7 days. Maybe we can tell them apart using these signatures. Or clinically more relevant, a cancerous cell versus a non-cancerous cells. Or also measuring the effects of drugs. So this is what I mean by modulation. If you add something or you change the microenvironment, how precisely can you tell its effects on the single cells? And with that, you can all go eat. I think we're done. So thank you for inviting me.